Hello everyone, how are you today? I hope you are doing well wherever you are. So through this video, I will invite you to check around and let observe what our routine is in Translational Research Laboratory of Shuang-Ho Hospital. So this is our main laboratory bench where we routinely do several type of assay. Such as here, we can see power supply, transfer tank, and gel holder for doing Western blot. And this is the drawer where we store our Western blot apparatus. This is also still part of our group main bench where we do other assay. As we could see here, there are some pipettes and reagent in this drawer. And also we have some refrigerated centrifuge machine and also water bath. And this is our flow cytometer machine, and this is our microscope and also the monitor to capture either immunohistochemistry or immunofluorescence staining section. And also this is the larger flow cytometer that is able to do more tasks such as cell sorting. Okay, we move to other room. This is our discussion room where we regularly have a lab meeting with other lab members and our professors. Here is the cryopreserve room where we have several freezers that may maintain the main temperature to -80 degrees Celsius, and also several drums to store the liquid nitrogen. I will let you have a look at what is it like inside. Okay, then we move to other part. These are our 4 degrees Celsius refrigerator where we store our new cell culture medium stock, including also some staining reagent and also some antibodies. This is another 4 degrees Celsius refrigerator where we store another space for the reagents and also new cell culture medium. And we also could see in the bottom of the refrigerator there is a shaker to do some incubation with primary antibodies. And this is our -20 degrees Celsius refrigerator where we store our antibodies and also some drug inhibitors and also some spatial reagents. So it is required to know which refrigerator we will need to use in order to well preserve our reagent. Move to other section. Here there are several PCR machines to make large number of copies of our DNA or RNA samples. There is also a nanodrop machine beside the PCR machine to quantify either DNA or RNA sample. And this is our fluorochem imagers to detect and capture the band intensity from Western blot membrane. Next to this machine, there is ELISA reader to read and detect either fluorescence, luminescence, or absorbance signal. And this is our automatic microscope that is capable to monitor our cell line samples real time, even up to several days or weeks. And this is our other group desk. And this is the chemical fume hood where we do our staining or any procedure that deals with volatile reagent. And in other room here we could see water bath when we need to warm our complete medium before being used into our cell sample. And there is also water distiller here to purify the water. And this is our regular refrigerator. In the freezer of the refrigerator, we could see bovine serum stocks, antibiotics, growth factor, and etc. And below, we could see our complete cell culture medium. And before we enter the cell culture room, we need to enter this blower chamber to sterilize ourselves. We move to other room. Here we will find the heater, and also we could see here several cylinders of carbon dioxide to maintain this CO2 condition of the cell incubator, and we could see the ice maker machine to provide us with the ice to maintain the temperature of the reagent, and also this is the heat oven to sterilize the pipette tip or dry some plate in several type of experiments. So we will move inside the cell culture room. Here in our laboratory there are around 5 incubators, including this one, and there are 5 laminar flow cabinets in total, including 2 cabinets in P2 culture room. And this is the microscope and the monitor to capture the image from the cell. In this laboratory, there is also the P2 cell culture room that offers with higher level of protection when we need to conduct our research to infectious pathogens, such as when we do cell transfection protocol using some virus. And here is my favorite laminar flow cabinet where I usually do the cell culture. And there is also the centrifuge machine when we want to make the cell pellets from our cell line sample. Okay, so here we go. Let's start our routine experiment. So at first, in this scene, I will conduct the cell cytotoxicity assay with my B-cell lymphoma cell lines that are scattered in 96 well plates. So before this scene, previously I have already treated all of the cells inside the well with some specific drug inhibitors and already incubated for 72 hours. Now, because the time was over after incubation, so I start to insert each of the well with staining solution that quantify how many cells that are viable in each well. So I will just make it faster. Okay, it's already complete. Then for the next experiment, I will do the cell culture procedure, but I need to check first my B-cell lymphoma cell line to know their confluence and also the medium condition. And to do so, I will check every plate under the microscope for their confluence. then next, I will return to the laminar hood and start to take routine care for the cell. So I will collect for each plate medium that contains cell line samples into the centrifuge tube. Then I just take the tube into the centrifuge machine to let them rotate for about 5 minutes. And this process will produce cell pellets. Start, and during that process I will prepare new plates for each cell sample and also fill the new plate with new complete medium. So basically, taking cell samples is just like treating human, because they also need nutrient from the medium and they also may produce waste to the medium, so we need to regularly change for the medium. Some of the cells will also die, and others will also undergo mitosis to divide. That's why we need to overlook our cell sample. So after the centrifugation, I just take out the supernatant and then reseed the cell pellet again. Some of the portion of the cell pellet I just reseed into the new plate and new cell culture medium. Okay, after that, I will check again the subculture cell under the microscope. And then I return back all of the new plate into the incubator, and we will meet again our cell sample for next 2 or 3 days, and done. However, after this, I will have some discussion with other lab member. I just discussed with my senior Indonesian student, Dr. Dharma, to solve the problem regarding how to generate scoring system for survival prediction in cancer using genes expression data. Okay, I think that's all for today. I hope you enjoyed this video. Please stay safe and keep healthy. Bye bye.